we are going to start right now. Maybe people will join in 5 minutes. I will remember, and we also will write an email after everything finishes with information, extra information. Maybe we can share the videos, the presentation, and also the recording. This is another session of Meet the Scientist from the project Global Excursion. I am Enrique Barra. I collaborate with the project. I am one of the developers of the ViSH platform. That is a social network to connect scientists and teachers. So today we are talking with two scientists from UCAM, University of Cambridge. Their specialty is about nanoscience, and they will introduce themselves because I'm not such an expert in that field. Hello, Malik, and can you introduce yourself?
Yes. So this is Harry who will start.
Okay. Well, I'm Harry. I'm a PhD student here at the University of Cambridge, and Mike is a postdoc. So we work partly at the Nanoscience Centre, shown on this slide. And today we're going to be talking about, in particular, one of the pieces of equipment that we use here, the Electron Microscope. A bit about how it works and what we use it to image and why those nanomaterials might be interesting.
Yep. So we're going to start with a short introduction, what we are interested in and why we are working on nanoscience. So as you all know, there are quite a lot of energy-related challenges nowadays. So the first one is energy conversion, for example, the conversion of sunlight into electricity. Then, of course, also the storage of energy, and also to improve the energy efficiency of different devices. And so there are different approaches science is going right now. So either you can start with a really complex, big approach, so you can come up with really new technologies such as fusion power, or what you could do is you could also use the existing technologies such as solar cells, batteries, capacitors, and catalysis for splitting water, and try to improve the materials that are used in these devices. So I would call this the chemistry approach because the aim is to synthesize new and novel materials. So, but our approach is even less complex because we're using existing technologies and existing materials. And so what we are trying to do is like we are trying to enhance the properties of these materials by nanostructuring them. And so the easy way to explain that is, so we are working with functional materials, but typically if this material is like bulk material, only a really small shell around the material is actually active material. And most of the bulk material isn't really contributing to the functionality of the device. For example, battery and catalysis and other devices we are working on, or solar cells, for example.
Because the processes that are important there are happening at the interface between the different materials. So it's only the surface that you're interested in.
And that's typically only a few nanometers. So what we are doing basically is we are nanostructuring the materials, and by doing so, we convert all the material that is used in the device into active material. And so this cartoon just illustrates that. And so yeah, that's what we're doing. And like often we use the electron microscope to then see what structures we have.
So this is just a photo here of the SEM, the electron microscope we have in the Nanoscience Centre. The SEM is because it's a scanning electron microscope rather than there's another sort of transmission electron microscope. We'll just talk through the various bits you can see in the photo. So on the right, you've got also the computers that we use to actually control the machine. The chamber housing the actual sort of sample that you want to image is in the middle of that block to the left. So you need it to be inside a vacuum chamber so that the electron beam doesn't get scattered or blocked by air. So it has to be under vacuum. The electronics controlling it all are housed in the big box underneath that chamber. On top of the chamber, you have the electron gun. So just something that accelerates the electrons and focuses them into a beam that you can use for imaging. And off to the left, you can see there's an X detector, so you can detect X-rays that might be given off by the samples during imaging.
Okay, so and in this specific microscope, we are using an in-lens detector. So as you can see, the electrons are coming top, are going through the this gun, and then are hitting the target or the sample at the bottom, and then the electrons are backscattered, or secondary electrons are scattered into the detector. And so this is an in-lens detector, and typically this gives a really high resolution and really nice SEM images. And so the electron beam, the incident beam, is interacting with the interaction volume. And as Harry mentioned before, there are two different kinds of SEMs, or no, sorry, two different kinds of electron microscopes. The one is a scanning electron microscope, and the second one is a transmission electron microscope. So the SEM looks at electrons that are backscattered. So typically the samples, you can basically look at any surface, while the TEM requires really extremely thin samples because the electron beam has to shoot through the specimen. And so in the SEM, what we are typically interested in are either the backscattered electrons. So these are the electrons that really are the ones that are in the beam and that are scattered. And then the secondary electrons, the ones we are looking at with this in-lens detector, are the electrons that are, so in the specimen, you have a lot of electrons sitting in the material, like surrounding the nucleus of the atoms. And these electrons are then kicked out of their shell and fly towards the detector. And these electrons are detected. So let's go back a step. Electrons have different energies depending on how fast you accelerate them, but it's different compared to light because we can see different colors because the different photons, so the different light particles, can have different energies, and depending on the energy or the wavelength, these particles or light particles our eye can see or notice that their energy difference, and that generates the color we see. But the SEM doesn't have this ability, so the SEM only sees whether there are electrons or whether there are no electrons. And so, sorry, the slides were just jumping around. So just imagine electrons can be either waves or particles. So if we look at like electron as a particle, just imagine you want to look at something like a big object and you're blind, and the only thing you can do is like you can take a tennis ball and throw the tennis ball at an object. And typically, so if you're throwing at a wall that is perpendicular to you, the ball will come straight back at you. And then you know, okay, the surface you're looking at, there is a surface, there's something away, and the topology is in a way that it's like perpendicular or to you. And so if the wall wouldn't be like a flat wall, the ball would jump into a different direction and you wouldn't come back to you. And that's actually what this SEM is doing. And so by scanning, because it's called a scanning electron microscope, it's scanning through the different areas. So it's scanning along lines and always detecting whether there are electrons coming back. And so that's what a scanning electron microscope is. So just coming back at how we prepare these nanostructures. So we start off with templates that are highly porous, and we use electrochemical deposition. So we apply a potential current, and then we deposit materials into these templates. And then in the second step, we remove the template and build these functional devices out of the materials we nanostructure. And let me just point out, this is a really versatile method, but the tricky bit actually is how we prepare these templates, because it's really hard to get three-dimensional nanostructures and then different materials with that. And in our lab, we use self-assembly, and this self-assembly happens in die blocker polymers, and we then use these nanostructures. So just to give you a short insight into block-of-polymer self-assembly, polymers are basically plastics, so that long chains of macromolecules, and if they just like water and oil, they demix and make different phases. And since we're not looking at homopolymers, but at diblockopolymers, so polymers with two chemically distinct blocks, the phase separation happens on the nanoscale. As you can see at the image on the bottom right, so each of these blockopolymers has one chain, one plastic chain, made out of these red and blue repeating units, and they then start to phase separate and make these red enriched and blue enriched phases, and these are then nanostructures, since the molecules are the length of them are on the nanoscale. So, and depending on the volume ratio of these two blocks, the blue and the red block, you can get different morphologies. And as you can see in this slide, for symmetric blocks, you get lamellae, and for really asymmetric blocks, you can either get cylinders or spheres. And in between, we get this really interesting gyroid morphology. And the thing why it's so interesting is because both the blue and the red phase are continuous. And this is really important if we want to make three-dimensional nanostructures, since as in the SEM image I've shown you here, so in the middle you see cylinders that were replicated, so the material is platinum, but as soon as you remove the template, they just collapse. And so you lose all the high surface area you had before. And just remember, we're doing this nanostructure because we want this high active surface area. And as you can see, surrounding the cylinders that collapse in the middle, we've got this really stable and robust gyroid morphology. And so, yeah, Enrique, I think you can play the gyroid animation now, the video. And Harry is going to tell you a bit about this gyroid morphology.
So this is a video about video gyroid animation, right?
Yes.
Yep. So when it starts, so this is when it plays. This is going to be a video. It's just a schematic showing the gyroid structure, so representing it, because it's quite hard to visualize.
Enrique, it's not playing.
Yes, I think the application is waiting for everybody to load the video, but I'm not sure.
Okay.
Because between you and me, it played.
No, it's not playing on our side.
Let me try again. Yes, it starts like loading. Let's wait.
No, Enrique, nothing is happening on our side. I think also the users, yeah, I guess just everybody press the start button, no?
Yes, press the start button. Don't you see it? I will try again.
No, Enrique, I think it's not working.
Okay, let me try again, because I think it's somebody connected with a small connection, a slow connection. So the application waits for everybody to load the video, and it takes a while.
Okay,
It
now
now
it's
works
working.
for me. Okay.
Okay. So yes, this is a sort of a schematic representation of this gyroid structure that we're talking about. So we've separated it because it's made up of those two different components, what we labeled the red and the blue part of the polymer. We've split this here in to show you the blue and the red network. And you can see then, if you etch out one of the phases, either the blue or the red, then you get left with this porous structure. So obviously the remaining one, the one you didn't etch out. And we use that porous structure as our template. And just to be clear, so this is a nanostructure because the size of these pores is around sort of 20 nanometers in size.
So yeah, if you take these unit cells and just put them together, you get a three-dimensional object like the prints we are using. Yeah, and we could just go back to the presentation, please.
To the presentation, right?
Yes.
I think it was slide 16. Wait, let me.
It's okay, I'll just go through it. So
I think I can move to 16 right now.
it always jumps back to the beginning.
Okay, there is a question in the chat. What do the blue lines stand for in the video? They mean.
You mean the surrounding lines? Oh, you mean the lines on the right-hand side or the black lines? So the black lines are the unit cell. They just show the smallest volume that makes up the whole structure. So it's a periodic structure. And if you just take one of these cubes and put it next to the next one, you will build up the whole structure. And so there were two. On the left-hand side, there was the red structure. On the right-hand side, the blue structure. And the blue structure fits into the red one. So the one is the majority phase, the red majority phase, and the blue network is the minority phase. So these are the two blocks. So you had the right, I'll try to go back. So like it's in this image at the bottom, you had this is like a cartoon of a polymer with the repeating units. So you have the red repeating units and the blue repeating units, and so the gyro structure, it fills the whole volume, and then we remove the blue structure. So and you can just draw these two different structures separately. So I hope this clarifies the question.
Yes, and this is all nanometers, right?
That happens on the nanometer scale, yes.
Nanometer scale, yeah. Oh.
So,
okay, so on this slide that we're showing now, again, we've shown in like this cartoon of the red part of the polymer. So this is when you remove the blue bit and get left with the red porous part. And around it are images that we've taken then using the electron microscope. So as Mike was before, you don't see any color on these. They're just grayscale because you're just detecting the electrons. Well, they have wavelengths, you can't really distinguish them.
So you only see how many electrons are reflected to the detector. So if there are a lot of electrons reflected, you get a white spot, and if there are like fewer electrons reflected, you will get a darker spot. And
so that's
yes,
why.
and
So, that's
scanning,
why where you get the colors. Not the colors. That's black and white. So there is no color. It's just in black and white.
No,
right?
they're grayscale.
It's grayscale, right?
Yes.
Depending
So
on the
if you
number of
detect a lot of
electrons that are reflected, right? Okay.
If you detect a lot of electrons, you will get a white or a brighter spot. And if you detect no electrons, you will get a black spot, basically. And so the electron beam is like reading through the whole image. And it's just like the old TVs where you had your TV screen and there was an electron beam at the back whiting the image all the time. So that's exactly what's happening in the electron microscope. You have a beam that reads across the sample, and for every spot where it hits the sample, the detector looks how many electrons are coming back to the detector.
So the brightness is dependent then upon the material that you're scanning and the topology it has, so its shape, and that's how we use it to measure the nanostructure.
And in this image, you see different cuts through the gyroid unit cell. So it's the same structure. It's just like different viewing angles, basically. And so there are small scale bars at the bottom right of the images, and they are 200 nanometers. So the structure is really, really small. And as you all know, a nanometer is like 1 billion of a meter. So 10 to the minus 9 meters. So it's extremely small. And Harry just mentioned, so a nanometer is the distance of a...
It's something like how far your hair grows in about a second. So just to give you a picture of how small this is.
Yeah.
So it's
Really, really small. Yeah.
So, as we said, so this is the red structure after removing the blue polymer block. And so the next step is we replicate this structure with functional materials. In this image, you see this is nickel or nickel oxide. So we can prepare a lot of different materials with this nanostructure. And as you can imagine, the surface area is extremely high. So this material is highly active. And in the middle, you can see this double wave pattern, which is like, and just shows how periodic this structure actually is. And this is after replicating the template and then removing the template. And then we make the SEM images.
So just to make clear that the point with the replication is that we can use the same template using exactly the same polymers, and we just deposit different materials within that.
Okay, Enrique, could you play one of the two gyroid videos, please?
Which one? Video
Doesn't
04?
matter. Start with number 4.
Okay. Let's hope now it plays quicker.
So, are we supposed to? No. So this is now a video where we actually, this is our SEM, where we zoom into a sample with some gyroids. And so at the bottom left, you can see the scale bar, how it slowly changes while we are zooming in. And so right now it's 20 micrometers, which is micrometer is 10 to the minus 6 meters. So already pretty small. I think 20 micrometers is roughly the diameter of the hair, right? Yeah. So
let me start again with it because I think the same as before, it didn't load it well for everybody.
So yes, again, we are slowly zooming into the sample.
It is starting to zoom. Wait. This happens because it plays to all the people that is watching this session at the same time.
So while the video is loading, are there any questions? So if you just type the question in the chat, we will try to answer it.
So what we're going to see is that you use a computer program just to monitorize what is happening in the electron microscope, right? There is a Windows application.
Sorry?
There is a Windows application that
yes,
you have.
Yeah, that's a Windows application. So it's running on a normal PC, but there's a lot of electronics also in the electron microscope. Yeah, I mean, you mean so there's a question from Barbara, and she wants to have some examples on how this technology is applied in industry. So, are you talking about electron microscopes or the structure we are preparing? So, because electron microscopes, so we have like here in which we have at least 20 or 30 electron microscopes, and yes, the structure. So this is actually pretty novel research we are doing on this structure, but there are already a lot of nanoparticles mainly. So nanoparticles are like more one-dimensional structures rather than like these three-dimensional structures. And nanoparticles are used in a lot of different applications. For example, in white paint on your walls, there are titanium oxide nanoparticles that give the white color. And then there are a lot of, for example, in toothpaste, there are titanium or titanium oxide nanoparticles, and they just help to clean the teeth. So there are a lot of applications in these structures. And so let's come back to the video.
The video is playing, I think, right now.
We are zooming into this gyroid structure, and as you can see, I think it's really, really beautiful. And so you see, really, it's highly porous and actually pretty stable. And so actually there are two networks, and both of these networks are fully self-supporting and stable. And Yeah, this is now a really high magnification. So the strut diameter is roughly 10 nanometers.
Yeah.
And so it's extremely small. And here you can also see how the beam, the electron beam, is slowly scanning the sample, and
Yes,
by
it
doing so,
comes
it's
and goes.
generating the image.
That's like in a television, right?
Yes, it's exactly the same principle, basically.
You can also see the regularity in the structure. So it's you can take that unit cell and just stack them next to each other, and it's just a repeating unit, and that can make it a good test case, seeing how nanostructure in general might affect the properties of various devices that use it.
Okay, Enrique, can you quickly also load the second gyroid video?
Okay.
So this is also, so the video we looked at before is like a full film of these gyroid structured materials, while the next video shows like a nucleation and growth process. So doing electrochemistry, we only have a low density of the nucleation points, and then we get these actually nanoparticles that have an internal gyroid structure. And which is pretty funny is we also get facets on these particles, and the facets arise from the crystallinity of the template rather from the crystallinity of the underlying atoms. And actually that's pretty unique, because typically if you see facets, you would think that the material is crystalline, but in this case the material is polycrystalline, and the high order of the template gives rise to these facets.
Because as you grow it, it wants to grow in different directions at different speeds, basically.
Yeah. So again, as we zoom into this particle, you see the nanostructure and this really high periodic, beautiful structure. And I forgot to mention, so in the next few minutes, we're going to talk about butterflies and how butterflies generate color with structural colors. And actually, there are some butterflies who also have this gyroid morphology as a photonic crystal that generates a green color, for example. So you can also find this structure in nature. So I think, Enrique, we can stop the video here and just quickly load the video of the nickel oxide device.
Okay, so people are asking what might be an application of these structures. So here we've got what's called an electrochromic display. So as you can change the color of the material by applying an electrical current to it. And that's what we're hoping to show in this video here. If I wait for it to load. So we've printed a crest, the university crest, is the shape you're able to see in the middle, and basically within that volume, it's all nanostructures with this gyroid structure. And the advantage of having that porous structure there is that it can very quickly change its color state. So that's how the nanostructure is helping this with the switching time between the two colors.
And it also improves the contrast, so the color contrast of the material.
So hopefully when this video loads, you'll be able to see as you apply the current by pressing the two buttons, the crest will become dark and light.
It's not loading. I don't know. Maybe I can try again. Because what we see here is about new display. Let's wait.
So I guess so I think the videos are going to be online on YouTube, right? So you can also have a look at them.
Yes, we will send after this session. We will send a report with all the content in case anyone wants to use with their pupils, with their students.
So are there any further questions?
I have a question about how many this kind of microscopes. How many of them do you have? Do you use them a lot, or do you have to ask for permission to use them, or are they freely available for you?
There are quite a lot of these SEMs around. So they are the cheapest ones start at roughly 150,000 pounds, or like roughly 180,000 euros, and then they become pretty expensive pretty quickly. So the higher the resolution, the higher the cost. So
Yeah.
for example, these transmission electron microscopes, they can cost up to a few million pounds or euros. So they're really expensive. But here in Cambridge, we have quite a lot of them. So we have one here in the building, actually two SEMs, and then the physics department has another 4 or 5, and also TM. So there are quite a lot of these electron microscopes around, and so we use them quite frequently because there are not many different techniques how to look at the samples. It's basically only this SEM where we can really zoom into the structure and see the nanostructure, because with optical microscopes, there's really a limit. You can only like look at structures that are around like micron size, and you really need really good optical microscopes to like zoom in any further. And so the resolution is just limited actually by the size of the light particle, if you want to say it like that. So as you know, light can also be described as a wavelength, and the wavelength then determines what the smallest structure is you can look at. And visible light is roughly between 400 nanometers, the wavelength, and 800 nanometers. And so like you can't go smaller than that. And as soon as you want to look at a structure that's smaller than, let's say, a micron, you have to use an electron microscope. And so, yeah, we have to book these microscopes. We have a online spreadsheet to book them, and then we can just use them.
They're in pretty constant use, and actually, they're actually very easy to use. So you just load your sample, and I mean, as you saw before, the video that we were playing was taken directly from someone using the electron microscope. It's just a program you use on the computer, so it's very quick and easy actually now to use it as well.
So Enrique, let's just
being
skip this video.
so expensive equipment, is it available for everybody in the department, in your group, or you have to have a a hierarchy or a specific rank? You have to ask anybody to let.
So yeah, for example, I'm a manager of the microscope here, and Harry is an instructor, and so we work together. So if there are new people coming who want to use the microscope, we train them, and then we give them an account, and so they can book it. And typically, for example, for commercial users, they have to pay £100 an hour to use the microscope, while students have to pay £25 an hour. So so also like commercial users can come and use the equipment. Of course, they have to get proper training because, as I mentioned before, these machines are really expensive, and it's easy to damage them. So we need a proper training, and then people are allowed to use them on their own. Enrique, let's skip this video, please.
Okay.
Let's carry on with the presentation.
I will come back to the presentation. Let me go to slide 20. No. 20. No.
Enrique, I'll just go to the slide. Could you send the links of the YouTube videos?
Yes.
So the next thing we want to talk about is butterflies. Harry, maybe you start.
Yeah. So whereas we're trying to make these structures for technological applications, you can also find structures on the nanoscale in nature. So just as Mike was saying, it's quite hard to use an optical microscope using light to resolve details beneath the wavelength of light, so around 500 nanometers. If you have structure on that length scale, and specifically periodic, so repeating structure with that sort of length scale, then you get what's called structural color. So this is different from sort of pigments, which is chemical color, whereby the colored molecules are absorbing light. This is more reflection of light and interference, because light is, or can be thought of as a wave. You can get interference between the peaks and crests of the light.
So basically, which is really important, that is that the material these structural colors are made of is transparent. So if you would have a bulk material, it's just transparent. There's no intrinsic color to the material. And just by structuring on the same length scale as the wavelength of light gives rise to interaction of the material with the wavelength, with the light, and then it creates the color. So this is the morpho butterfly. We are going to talk about this in a moment. And on the bottom right, you see soap bubbles. And the colors you see on soap bubbles are also structural colors because it's called thin films interference. And so the soap and the water are transparent. So if you just look at the soap water solution, you don't see any colors. But as soon as you make like a really, really thin film out of these out of these materials, you suddenly see a color effect. And the thing is, on the bubbles, because there's no internal nanostructure, it's just reflection on this thin film. It's not one predominant color like in the butterfly. So, and that's why we are interested in using nanostructures to make color. And Harry will now explain to you the difference between pigment colors and structural colors.
Okay, so this is similar to what I was talking before. So pigment colors, this is when you have molecules that are absorbing a certain part of the visible spectrum, so certain wavelengths are absorbed and the rest is scattered back, and that would be the color that you then see for that material. Whereas with structural color, if you have, so white light is made up of light with lots of different wavelengths, and because of the structure, you as you reflect off the different layers shown in this diagram, the distance that it travels between successive layers is going to correspond to some of those wavelengths in the spread you have in white light and not others. So you get strong reflection at some wavelengths and not at others. And that gives you the color, because color is just the wavelength or the energy of the light.
And actually, the amazing thing about structured colors compared to pigments is like typically, I mean, if you have clothes and you leave them outside in the sunlight, after some time, the pigments will degrade and you will lose the nice vibrant colors. And this is not the case with structural colors because as long as the structure is intact, you will have this really, really nice iridescent and structured color effect. So these butterflies, also if you them for years in like in a cupboard, they will still look really, really like bluish or and vibrant. So and this is also a huge difference between pigments and structural colors. So here's one example of the Urania butterfly. And as you can see, it has different areas on the wings. So the scales are made of this multi-layer structure and create really, I would say, gold color and also purple color, or yes, at the bottom. And this is just thin film interference, like multi-layer stack, as you can see on the C, and this generates these colors. So there are absolutely no pigments involved in generating these colors. And the material the structures are made out of is chitin, which is transparent again. And so so in the middle, in B, you see a TEM, so transmission electron microscope image, and the darker areas are the chitin, and the brighter areas are just air. So there's a contrast between the chitin, which has a higher refractive index than air, and then the light starts to interact with that, and you get this multi-layer interference that then creates the color. So this is the next butterfly, the blue butterfly. It's called morpho butterfly, and it's really worth, like you can buy these butterflies over eBay. And so we also have a video of this when zooming into the structure, and I think we can try to load that, Enrique, the morpho butterfly video.
Let's wait to see if it
So
loads now.
this butterfly lives, I think, in South America, and also in definitely in South America. And people claim that if you fly with a plane over the rainforest, you can really spot this butterfly like a mile, like when you're flying a mile over the rainforest, because the color is really so vibrant and so yeah, just eye-catching. And these structural colors are often iridescent. So if you have this butterfly and you move it around, it's like really shiny, and so they're really amazing. And again, there are no pigments involved in these colors. So yes, so Barbara had a question is if we can see the color in the dark, or whether they would be transparent. So as I said, this material, we are also using polymers or plastic, like normal polystyrene plastic. It's like a lot of Tupperware and stuff is made of polystyrene, and it's fully transparent. And as soon as you start to nanostructure it on this really small length scales, you suddenly get like really nice colors. And so it's more, it doesn't have to do anything with the dark, because if it's really dark, so there's no light, you wouldn't see anything. But if you really, so what we tend to do, so if you have one of these butterflies, so not, so wait, but I have to start from the beginning. Not all butterflies have structural colors. It's only if, like a subset of butterflies who have structural colors. There are also a lot of butterflies who have pigments, but also here in the UK, there are some butterflies, or in Europe, that have structural colors. And the way to test whether they have structural colors is if you put a drop of ethanol, so alcohol, or also water onto them, but ethanol is better because that wets the structure. And excuse me, as soon as the ethanol infiltrates these structures, it's basically you lose the contrast between the air and the chitin. So the reflective index contrast is lost, and then also the butterfly wing scales suddenly become transparent. So it's really more about nanostructure, whether it's dark or bright. It's about having this contrast between the two materials. And as soon as you would like like squeeze the structure together and you lose the structure, you also lose the color. So I think that's a good way to explain it. So whenever you like, so as I mentioned, you can buy these butterflies online, and if you then take just a drop of alcohol and put it on them, it typically the color just goes away. They're pretty transparent, and as soon as the alcohol evaporates, the colors restore. So it's pretty amazing.
Okay, the video seems not to be playing. I think we can continue with the presentation, and in the end, I will try another possibility of sharing the video.
Let me just say 2 words to this video. So,
Okay.
First, you zoom into this morpho butterfly structure with the optical microscope, and then at some point, so that's when you see this blue and greenish color. And as soon as they zoom in even further, they jump from the optical microscope to the scanning electron microscope, and then the color scale is it's a gray scale, and rather than colors. And there you then can really see this nanostructure that's responsible for this iridescent color. Okay, let's go back to the presentation, please.
Okay. I will try another possibility of sharing my screen in the end, but that's quite experimental, so I will try it in the end, just in case. So let me move.
Enrique, I'm moving, it's okay.
Ah, okay, okay.
It
You
always
can write the number
at the beginning.
Ah, okay.
So we talked
But
about
you can write the number.
right?
Yeah,
And
now.
so this is another so TM cross section of the butterfly wing scale, and we call this a Christmas tree structure. So, and as I said before, this is the one that's responsible for the color. And again, the dark material is made out of chitin, and the brighter material is basically air. And at the bottom is another SEM image of the samples of one of our attempts to replicate this butterfly structure in the lab, because we are just interested in replicating these structures, because they would have a lot of different applications in optics, for example. And yeah, for example, if you want to color your car in a really nice shiny iridescent color, that would be a possibility to use structural butterfly. Again, the green areas are made out of structural colors, and actually these are concavities with a multilayer. And Harry is going to talk a bit about how we can prepare these concavities in the lab and also generate like really vibrant colors in the lab. But before that, we're just going to briefly talk about other animals that generate their colors using structural colors and nanoscale. Yeah, just mention.
Okay, so this is this Japanese jewel beetle has obviously got more structural color again.
Yeah, and so for example, some of these colors really look like gold, so it's pretty amazing. And there are also other beetles. This another Japanese shoe beetle. Then there are, for example, birds that have structural colors. Fish have structural colors. Some of them, and always when the color is really vibrant and iridescent, the colors are mainly made out of structures rather than pigments. So
okay, so if this video works, this is video now, so it's still staying in sort of the animal kingdom, but rather than looking at structural color, what we're imaging here is the eye of a fly. Now, flies have compound lenses rather than the sort of single lens that we do. So their eye is just basically made up of these long rods that basically each rod cuts out a little bit of solid angle that the fly might be looking at, and so it channels from only one certain direction, and it uses lots of these different rods then in the array that we're going to be imaging here to build up the image that the fly sees.
So, I mean, the video isn't working for us again,
No,
but at the
let me
beginning
try
of
the
the
other
video,
possibility.
you just see the sample stage where we have a fly that was spotted with some gold in order to making it conducting, because as you can imagine, we are talking about the electron microscope, and the electron beam tends to charge up the samples, and then the image quality isn't good anymore. So we use a really thin film of gold or another noble metal to coat this specimen and then have a look at them. And at the top, just at the beginning of the video, you see this electron gun. We showed the schematic like really at the beginning of the presentation. So I guess like the video isn't working for us again, so I think we should just skip it and
yes, I think in the end I will try the other possibility. I think.
Okay, I'll just quickly jump back to the. So.
Okay, so as another example of structural color in nature, it's not just in animals that you can find it, but you can also find it in minerals. So here, this is a picture of an opal. You can see there's patches of very brightly colored regions again. And this is structural color. So to the bottom right is another cartoon showing what's going on here. Basically, inside the opal, you've got silica spheres that are all of a very similar diameter, and they stack together in this sort of structure that you can see. So it's another ordered nanoscale structure, and because the spheres are on the length scale of light, you get these different colors, and it's as you would hold the stone in your hand, if you were to look at it from different directions, the colors would change as you'd get different interference patterns.
And again, which is really important, that the silica, so which is silicon oxide, is transparent. So the material, the intrinsic optical properties of the material, the bulk material, is that it's really transparent to light. but just by ordering it on this nano length scale, you get this strong iridescent color or opal effect.
So only by building it in the structure do you get the colors that we're seeing. So that's a natural opal, but we can try and fabricate them synthetically as well, make artificial opals. So this is just quickly showing how we might do that. In the vial that's shown to the right, you can see these. In fact, we use polystyrene rather than silica, but these polystyrene spheres are suspended in water, and you get this fluid in the vial, which is a colloid. So it's just these polystyrene spheres suspended in water, and they look white here because they're scattering light. Again, it's kind of like a structural color, but you haven't got a periodic fixed structure. They're just sort of free in the liquid, and that's why it looks white. And in fact, it's similar to milk. So milk is another colloid, and that looks white for the same reason. If you put a drop of this colloid solution, these spheres in solution, and dry them by dragging a blade across the substrate that you put the drop on, which is what I've shown in this image here, then the evaporation of the water that happens at that small gap between the blade and the substrate, at that gap, the water evaporates and leaves behind just the polystyrene spheres, and then in fact towards that gap as the water evaporates by surface tension, and this can induce the stacking that orders them into that opal structure. Hopefully now if the video works, I don't know, we'll give it another try maybe. We can show you first of all an optical microscope video of us. Yeah, this should be it. Depositing the spheres. So if you're playing, you should hopefully see the blade is the big dark bit on the left of the screen, and as that moves away to the left, on the right, you can see the polystyrene spheres are deposited from the solution, and just
I will
as the
reconnect.
water dries, it leaves behind these polystyrene spheres on your substrate, but with the regular structure that is the opal structure.
So Enrique just left the room. I hope he'll be back in a second because he has to switch back to the PowerPoint presentation.
Okay, so I think Enrique is just trying to reconnect. Hopefully the videos might play then.
So again, these colloids are made out of polystyrene, and polystyrene is basically like the plastic we use like almost everywhere. So, and this plastic is really transparent, and just by like ordering it in this highly periodic way, these colloidal films then really produce a really nice structural color. So I think now Enrique is trying to.
Yes.
Enrique, I think so. Dia doesn't hear us, so I think we might just also quickly try to reconnect.
Yeah, I'm done. Hello? I'm just sharing my screen so anyone can see the video. Hello? No, eso no es buena idea.
So I hope you hear us. So
Yes, I hear you.
yes, okay. So yeah, in this video we just assembled the monolayer of these colloids, and there are some defects. Some of the colloids are slightly too thick, so they're not really monodispersive, and then this gives rise to some of the defects in this colloidal crystal. So
maybe I can try with the other video.
The SEM video.
The SEM, yeah.
Yeah.
Let me, because I have to share it in my screen.
Okay, so what we were showing before was an optical microscope video of the deposition of the colloid, using actually quite big colloid particles, quite big polystyrene spheres that you could see it. But you can go down to small nanoscales, even down to 200 or maybe 100 nanometers, and then you'd need the electron microscope again to image it. So if this video plays, that's what we're showing now. Again, you can see this is the sample loaded on the stage inside that vacuum chamber, and it's sitting underneath the electron gun, which is that cone at the top of the screen. So I don't know if the video is playing underneath this.
Yes, you should see now the SEM video. SEM.
Okay. So in a minute it should switch from. Oh yeah. So now we're looking at the image that the electron beam is seeing.
Yeah.
So that's the edge of the sample you can see, and this is at its most zoomed out. So if we slowly zoom into the sample, I think the video is paused for the moment, Enrique. It is for us anyway. Yeah. Okay.
Yes, it's the frame rate that is quite slow. Just to know, I will send the videos to everybody. And with these explanations, I think they will be able to understand what is explained there.
Okay.
And the examples.
In that case, just a quick explanation of what you would see when you see it.
So I will stop sharing. I will try with the YouTube again, just in case, but. Yeah. Lydia, okay, just to finish, I have also more questions. How many of you are working there? Do you have a lot of projects? Are they most research projects?
So there's lots of people working here. I would guess must be at least 100 using this nanoscience sensor. It's a variety of different people. So there's PhD students working here. We have master's students, undergrads from the University of Cambridge. There's also commercial users. So as Mike was saying, various companies who are based on the same site here at Cambridge, and they use the same equipment. Seeing as it's so expensive and can take a lot of expertise to use, it makes sense to have everyone in the same place using the same equipment.
So, and yeah, I mean, we are working on, also Harry and I are working on a lot of different projects. I mean, we're working on like these structural colors. We are working on solar cells, on batteries, on supercapacitors. So,
but are research projects, right?
Yeah.
Or you also have some commercial projects. You know, I plus D, so sometimes development, sometimes investigation, innovation. So it is mainly innovation because this is quite new technology, or do you have also commercial projects, development?
It always depends. Like the research we are doing is pretty fundamental right now. And so I would say it's more on the fundamental research, and still a long way down the road before commercializing these products. And so yes,
but there are companies who will use the research they're doing. You know, it might be basic science that does end up being used, and we also have companies that have spun off of work that's being done here.
Yeah. So for example, Barbara had a question whether we collaborate with the industry, and yes. So for example, my PhD was funded by Nokia, and there's a research center of Nokia here in Cambridge, and really close by, and we were working closely together with Nokia to develop new like nanostructures for cell phones and stuff like that. So yes, there's a really like strong tie between industry and the university. So for example, there are a lot of big oil companies who are interested in these colloids we were talking about earlier, because they need them to get the oil more fluid when they pump it out of the ground and stuff like that. So yes, there's a really strong collaboration happening with the industry.
Uh-huh. Because you said you also had the possibility for commercial people to go and use the microscopes, right? They have to pay a fee, but they
Yes,
can use them.
and as I said before, so commercial users have to pay around £100 an hour, so that's roughly 120 euros an hour to use the SEM. But then if you see how much it costs to buy a system like that, so you quickly
Uh-huh.
talk about
Yeah, it's
so
indeed it works because 120 euros is not so much
yeah,
if the equipment costs several millions.
that's right. So and you also need people who are servicing these big equipments, and so yeah, and they also need a lot of electricity. So um, you just it makes sense to share these facilities with companies as well.
And to answer Lydia's question, so PhD students are using this equipment all the time, and there is definitely opportunity for undergraduate students. They do short projects working with us in the lab, so students would use this equipment too. Yeah.
Mhm. And about the labs, have you ever tried schools that have? Do they ever go there? And like in an excursion, they go there with the teachers and you show the equipment, and
Yeah,
you
yeah,
have
we do.
that kind of activities.
So here in Cambridge, it's called Physics at Work, and there are
Uh-huh.
typically a few hundred school children coming here and having a look at the lab, and it goes a, like 4 or 5 days where children are allowed to come here and have a look at our labs and facilities.
Uh-huh.
And so yeah, I think that's definitely possible.
Yeah,
And
that's cool.
the students here also go and visit school classes and give presentations. And yeah, so I think there's a lot of interesting stuff to see, like with these structural colors, with SEMs. We have a big clean room, so where the air is filtered and there are hardly any dust particles in the air. And because we need that, because as you can imagine, dust is actually pretty big if you look at it under the microscope. So dust is our enemy, and that's why we have like huge clean rooms where we then have to wear overall gloves and stuff like that. And so it's
Mhm.
like great fun to work in these kind of labs, and a lot of chemicals standing around and a lot of
Yeah.
big machines. So it's really interesting to come and visit us.
Yeah, I think students or young people are also always very interested in about what physics and chemics do in their daily work because they don't know. They only will know what they see on television, or they watch on the television. So they get quite interested.
So
So
are there any
if
further questions or?
yes, if there is another question, if not, sorry for the video's problem. There must be one slow connection that doesn't get the video and the application waits for everybody to load, so everybody will get no video. But I will send a report with everything, also the recording of the session, all the presentation, and my email in case you have any doubt. Just to remember the people that are now connected, kind of experiments can be done inside the platform, inside ViSH Hub, and this tool is for free for everybody, the MashmeTV. It can be used, so if a teacher wants to use it to connect with his pupils or with other scientists or this kind of experiments, you are welcome. So thank you very much, Mike and Harry.
Welcome.
Thank you very much and see you. And
Bye.
thank you everybody in the chat.
See you.
Bye.
Bye. Hope you enjoyed it.